Hello everyone. Science Spot is a podcast that shines a spotlight on the latest scientific technology from Japan. Your host is Ren from Science Talk. So today's topic is tooth. Losing a tooth can be tough, right? Whether it's from cavities, gum disease, or an accident, current solution like dentures, bridges, or implants are functional. But they are still not quite like having your natural teeth. The dream of regenerating an entire tooth is a major research goal worldwide. Scientists are making progress in growing tooth germs in labs and transplanting them. However, there's a big challenge. We don't yet have sufficient technology to precisely control the shape, especially the length and the structure of the regenerated tooth roots. The tooth root is incredibly important. It anchors the tooth firmly in the jawbone and supports the immense forces of chewing. It also contains the apical foramen, a small opening for nerves and blood vessels, essentially the tooth's lifeline. Without a properly formed root, a regenerated tooth won't be stable or fully functional. Past efforts have included using 3D scaffolds to guide shape and directly manipulating gene expression in tooth-forming cells, but a more fundamental and precise method for root control was needed. That brings us to today's exciting news, a discovery by a joint research group from Tohoku University in Japan and the Mayo Clinic in the US. They've found the epigenetics mechanism that adjusts gene function without changing the DNA sequence, control calcification in dentin and cementum, and thus tooth root formation. Let's quickly define epigenetics. Imagine DNA as a vast recipe book. Epigenetics doesn't change the recipes themselves, it means the DNA sequence, but rather determines which recipes are used or how often. It's like putting sticky notes on certain recipe pages to highlight them or closing others to indicate they are not needed right now. This on or off switch for genes is crucial for cell differentiation and function. Central to this research is a specific enzyme involved in epigenetics, histone deacetylase, or HDAC. What is HDAC? HDACs are enzymes that remove the acetyl group from histones, which are proteins around which DNA is wound. And think of DNA as a long thread and the histones as spools. When DNA is tightly wound around these spools, it's harder for the cell to read to genes on that section of DNA, effectively turning them off. HDACs make the DNA wound more tightly. In this study, HDAC3 was the specific enzyme of interest. The research team, led by Dr. Nibe and Professor Egusa from Tohoku University and Professor Jennifer from Mayo Clinic, focused on HDAC3's role. They created genetically modified mice called KO mice, where HDAC3 was absent in cells expressing ostrix. Ostrix is a gene crucial for bone, dentin, and cementum formation. Then they analyzed the teeth of these mice. The results were remarkable. Compared to wild-type mice, the knockout mouse exhibited significantly shorter molar roots. Additionally, the apical foramen, the hole at the roof tip for nerves and blood vessels, closed prematurely in these mice. This strongly suggests that HDAC3 is essential for proper tooth root growth and maintaining openness of the apical foramen. The researchers also performed cell-level experiments. They treated cement blasts cells that form the cementum on the root surface with the HDAC3 inhibitor drug. They found that this treatment suppressed the expression of calcification-related genes and inhibited actual calcification by the cement blast. This directly demonstrates HDAC3's involvement in the calcification process of cementum, a hard tissue of the tooth. This experiments provides clear scientific evidence that HDAC3, through epigenetic mechanisms, controls critical process determining tooth root morphology, including root length, apical foramen formation, and cementum calcification. This research offers immense hope for future tooth regeneration therapies. It opens up new possibilities for controlling the morphology of regenerated tooth roots using epigenetics. By precisely adjusting HDAC3 activity, perhaps with medication, we could potentially control the length of regenerated tooth roots, preventing them from overgrowing and guiding them to form the ideal shape. This is a huge step toward making regenerative dentistry a reality. That's all for today's SciencePod. I'd love for you to listen to the podcast and post your notes and thoughts with the hashtag SciencePod. Thank you for listening and see you next time.